Transcription. A, an RNA comparison. DNA and RNA, both being nucleic acids, are mostly the same, but with some important key differences. You can see visually the differences here between DNA on the left and RNA on the right, and we'll take a look on the next slide at what those differences are more closely. This table outlines the major differences between DNA and RNA. DNA uses the bases A, T, C, and G. RNA, A, U, C, and G. The differing base is that DNA contains thymine, whereas RNA contains uracil. The base pairings are A with T, C with G, and in RNA, A with U. DNA contains the sugar deoxyribose. RNA is ribose. The backbone structure for DNA is sugar, the deoxyribose sugar and phosphate. The sugar phosphate backbone in RNA is ribose and phosphate. DNA is a double-stranded helix. RNA is a single strand. DNA is more stable due to the double helix. RNA is less stable due to the single strand. Transcription. Transcription is the process by which RNA strands are synthesized from DNA strands. Transcribe means to write out or rewrite. The process of making RNA from DNA is called transcription because the DNA sequence of nucleotides is rewritten into the RNA sequence of nucleotides. The RNA sequence of nucleotides will match the DNA sequence with the exception of U, uracil, replacing T, thymine. The process of transcription is similar to that of DNA replication. This is the first step in the transport of genetic information contained in DNA to the cell where the information will be used to build proteins for the cell's use. DNA has two strands. The template strand is used as the template for the enzyme RNA polymerase that will create the mRNA molecule. mRNA will take the information out of the nucleus where it can be used to create proteins. The non-template coding strand is complementary to the template strand and will match the mRNA molecule with the exception of T, thymine, being replaced by U, uracil. Genes. The segment of DNA needed to make a specific protein is called a gene. The genes reside on the non-template strand. DNA is a good archive for genetic information since the bases are protected on the inside of the helix. But this information has to get out to the cell. Transcription initiation. RNA polymerase attaches to the promoter region on the template strand of the DNA at the beginning of the gene that it wants to copy. You can see the promoter region here on this length of the gene where RNA will attach. The promoter is a DNA sequence that the RNA polymerase recognizes. This is a group of accessory proteins and positive transcription factors. Transcription DNA strands. Here's the important things to note. Does the strand contain the gene for the non-template strand? Yes. But the template strand is the one that the RNA polymerase attaches to and the one that gets transcribed. So think about this. As the template strand gets transcribed, it will match the non-template strand exactly with the substitution of uracil for thymine. So what we are copying into mRNA is the gene itself. Transcription elongation. RNA polymerase here runs down the DNA template strand, reading the bases and bringing in the new RNA nucleotides with the proper complementary bases. You can see that in blue, the new mRNA. As the RNA polymerase runs down the DNA, it unwinds the DNA. The RNA molecule that's being created in blue here will be an exact copy of the non-template DNA strand, this one up here. Of course, with uracil being replaced for thiamine. Transcription termination. RNA polymerase gets to a sequence in the DNA called a termination sequence. This sequence signals the RNA polymerase to stop transcription. The RNA polymerase falls off the DNA. The new RNA strand separates from the DNA, and the DNA recoils into a helix. Just like DNA replication, the template strand is read starting from the 3' end, and RNA is made starting with a 5' end and moving in the 3' direction. So if this is our template strand, we start reading from the 3' end toward the 5' end, and we make our RNA then. First will be A, U, G, C, and so on. So we're making the mRNA in the 5' to 3' direction. DNA replication versus transcription. In DNA replication, 2 new double-stranded DNA molecules are produced, whereas in transcription, 1 new single-stranded RNA molecule is produced. In DNA replication, adenine from the parent strand bonds with thymine on the new daughter strand of DNA, whereas in transcription, adenine on the DNA strand will bond with uracil on the new RNA strand. In DNA replication, the whole DNA molecule is replicated, whereas in transcription, only the strand with the code for the gene is transcribed. In both cases, the synthesis occurs in the 5' to 3' direction.